Hello students, I hope you all have been doing well. Today we are going to discuss experiment number 7, and this is about the qualitative analysis of protein in a given sample. So so the what sample we are going to what the protein sample that we are going to analyze in the given sample is the peptones. So this is our objective, and for this purpose we have to utilize a series of tests from X means test number 1 to test number 12. So the first test for this purpose would be the Barrett test. First we would be and this test was is for the identification of peptide bond in the protein. So whenever we have to analyze any protein in a given sample, so for the analysis of any protein in the given sample, first we have to find out that if the means if the protein in the sample is having peptide bond, or then we have to find out then if the given sample is containing the amino acid. Then we have to find out then if the given sample is having the protein, and then which amino acid this protein is made up of, and either there will be the aromatic amino acid, either there will be the aldol or sulfur containing an amino acid, excuse me. And then we have to carry out the different test for the identification of the particular type of protein. For example, we perform the acetic acid test for the identification of casein in the protein, that is a milk protein, and we can perform the coagulation test for the identification of albumin in the given sample, and then we can perform the peptone test for the identification for peptone in the given sample. So why we are means doing these tests one by one, and then finally we have to perform the test to find out the peptone in the given sample. Actually, whenever we are having any sample in the protein, we don't know about this what type of protein it is. Means the protein is unknown. We will be having a sample of unknown protein, and we did not know about the what type of protein is present in the sample. So we have to qualitatively analyze uh, uh, the presence of protein in the sample, and the we have to analyze qualitatively that what type of a protein uh, present in the sample, if the protein is present in the sample or not. First thing is that, and then if the protein is present in the sample, then what type of protein it is, either it is albumin, either it is globulin, either it is casein, or either it is peptone. So for the presence for the identification of the presence of protein in the sample, we have to perform the peptide a test that can tell us the presence of a peptide bond, because the protein are made up of amino acid, and amino acid join together with a peptide bond. So if by performing a test, that is the Barrett test, we will find the presence of a peptide bond in the sample, so it will be indicating that we will be having some protein in our sample. But at this first stage we did not know, we do not know that what specific type of protein is present in our sample. So the first test for the qualitative analysis of any protein in the sample was to find out the presence of a protein in a sample, because they may be the unknown sample maybe contain DNA, maybe contain RNA, or maybe contain carbohydrate or lipid. So how we can identify that the unknown sample that we are having is the sample of the protein? So how we identify? We identify by performing the test that can give us the indication then that the peptide bond are present in the sample, and the other test that will identify that amino acid is the present in the sample. So um, so when we will be find out that peptide bond are present in the sample and the amino acid are present in the sample, so it is the indication that there is a that this will be the indication that the unknown sample is will be containing the protein, because we will find out first the peptide bond identification and then amino acid identification. So from by these two tests that we will these are two the preliminary tests, and by performing these two tests, one is the Barrett test and other is the Ninhardin test, if we would be find out in our sample that peptide bond and amino acid are present in the sample, so it will be the indication that this sample is containing the protein. So so then we will would perform another test that will means uh, indicate us the visual presentation of the sample, how it would be happen by precipitating means by the precipitation test. So what does mean by precipitation test means, if we will be having adding 2 ml of 2 ml of lead acetate in our sample, and we will be finding out the precipitate in the bottom of the test tube, so it will be the indication that that the protein is present in the unknown sample. How we can say that protein is present in the unknown sample, because previously we have performed these two tests, one is the Barrett test and other is the Ninhydrin test, and these two tests have indicated the presence of a peptide bond and presence of amino acid respectively in the sample. So if we previously know that what is the unknown sample we are having, it is containing the peptide bond, it is containing the amino acid. So then we perform the precipitation test and we will get the precipitate at the bottom of the means test tube. So it will be it means indication that this is the actually protein that is present in the unknown sample, because the lipid and the carbohydrate would not precipitate, only protein would be precipitate. So then this stage at test number 3 would be means actually identify the presence of protein in the sample. Now after that we will perform a confirmative test for more confirmation or more cross check if the sample is containing protein or not, or this whatever the precipitate we are having in the unknown solution, is this the is this the means actually protein or something as precipitate. So for this purpose we would be performing the as barrage test. So in this test we would be adding we should have to add 3 ml of as barrage reagent and then 3 ml of the sample. So we would be having means if there will be protein, so the precipitate will be remaining in this actual means condition, so this and will would not dissolve. So this was the indication that the either the this is the protein actually that are present in the unknown sample. So after the confirmation of the presence of protein in the unknown sample, we would be going to find out that what type of amino acid is made means the protein is made up of what type of amino acid. Either protein is containing the aromatic amino acid, either protein is containing the indole amino acid, either protein is containing the sulfur containing amino acid. So this was will be our next means goal. So for this purpose I am highlighting it with these tests for the identification of amino acid, that what type of amino acid are present in the unknown sample with the green. So for the confirmation of the presence of protein, what test we perform I am highlighting them in red. So our whole experiment would be consist on three part. One part was to find out either the protein is present in the sample, that will consist on this four tests, and the next part would be the either the protein is containing what type of amino acid. It would be part 2, and in that it will consist on three tests. So why we and then the last step or last part of our experiment would be the actually the finding out that actually what class of protein is present in the sample. Either it is a casein, either it is the milk protein, that is casein, either it is the albumin, that is the egg protein, or either these are the peptone. So I am highlighting them in the blue for your convenience, that you can understand that our each experiment would be consist on three part. One part for the identification of protein, that would be consist on four steps or four tests, and then other next for the identification of which amino acid is present, what type of an amino acid is present, it is would be consist on three tests, and then last one, that what type of a protein or what class of the protein is present in the unknown sample. So why question is this, why we have you have understand this, that why we will perform means step test 1 to 4 for the identification of protein in the sample. So why we are performing means these tests from 5 to 7 for the identification of which type of amino acid is present in the sample. Either protein is containing aromatic amino acid, either protein is containing indole amino acid, or sulfur containing amino acid. Why we are means we are keen to know about what type of amino acid or what class of amino acid are present in the protein. Why? Answer is that because the type of amino acid would correspond to the class of protein. Means if we are will be having aromatic amino acid in the sample, or if we will containing the for example, um, uh, we will be having after performing these three tests, if we will be finding out that our protein is containing the indole amino acid, so specific type of protein from means or in this casein, albumin, or peptone containing this sulfur indole containing amino acid. So means each protein from the next part of the experiment, means each of this protein, either casein, albumin, or peptone, uh, having uh, high percentage of one of the amino acid, one of the class of amino acid from this from these amino acid. For example, some type of amino acid are means found in abundant in casein protein, some type of particular type of amino acid found abundant in the albumin, and some particular type of amino acid found abundant in peptone. So the identification of particular class of amino acid would be means help us to find out the particular class of protein. So I hope you have understand why we need to find out the class of amino acid. Now the so for find out the what type of amino acid would present in the sample, we can perform the xanthoprotic test. It will indicate the presence of aromatic amino acid. We will then perform um, So if we perform this test, xanthoprotic test, and we find this test would be negative, so what we will be do, then we will again perform the Hopkino cold test. So if we find that in the sample, if this test would be positive, so it means there will be indole amino acid in the sample. Or if this test would be negative, then we will perform sulfur test. So we have to perform all three tests. Either means our sample would be containing the amino acid, for example aromatic, indole, or sulfur or not. But we have to perform because certainly one of the class of amino acid would be find in our sample. So after that we would go to find out the particular class of protein in the sample. So we will perform first the test for the casein, for the identification of casein, or to find out if the casein is present in the sample or not. For that purpose we will perform acetic acid test. And after, if this test would come positive, then it means the casein is present in the sample. But right now we are we our means our our excuse me, our aim to find out the peptone in the sample. So what we will do, we will perform this test, that is the test for peptone. So for that, what we have to do, we add 5 ml sample plus 1 ml acetic acid. So we would be having whatever observation we would be having, we write here, and then result will be right here. And then after that we have to perform the confirmatory test for the peptone. And for that would be having 2 ml of 5% NaOH and 2 drop of copper sulfate. So this would be the confirmatory test for the peptone. And then we you have to after that we have to write down the result and the discussion in the discussion section. But you have to keep remember that before performing this test number 11 and 12, we have to perform before performing these two tests, we have to must perform the acetic acid test and coagulation test and saturation test. Because if our aim is to find out, as the objective of our experiment is to um, qualitatively analyze the presence of a protein in the sample. So what protein? Peptone. So if the sample is containing the peptone, so it means that all these tests would come negative, and then only the test 11 and test 12 would come positive. So this was the indication. If all these tests will coming negative, so this is the will be one more indication that the sample is an unknown sample is containing the peptone. And for the confirmation, as or for the confirmation that yes, this is the peptone that are present in the sample, we would perform the peptone test, and then we reconfirm our result by this test number 12. So this was whole about the experiment number 7, and I hope you well, miss understand it. Thank you very much. Allah Hafiz.